I'm gonna let John take it away.
Hey, good afternoon. Happy Friday. I'm not sure how many folks are in the audience or how many new folks are in the audience. But my name is John Kominoski. For those of you who haven't met me yet, I'm the person here in the center of the screen. I'm the lead principal investigator for the FCE-LTR, and we're in our 25th anniversary. We had that celebration last May that many people were in attendance at. And as you probably heard, or if you haven't heard, it's been announced officially by FIU and then also in our FCE newsletter that our fifth iteration of the LTER was renewed. So FCE 5 has started. And these are the co-PIs. I want to give a call out to um, the co-PIs that are on FC5. Dan Gann is a spatial ecologist. Tiffany Troxler, she's a long-term collaborator of the FCLTR. She's an ecosystem ecologist in Earth and Environment, doing a lot of work on ecosystem assessment and adaptation. Rolando Santos, who's at the Biscayne Bay campus in biology. He does a lot of seascape work and looking at, collaborates with James. I'll show a few slides of some of the data from Rollo's group. Also partners with Dr. Jennifer Rehage, James's major professor. She does a lot of work with exchange and movement of fishes and the trophic consequences in response to restoration and how that's changing fish movement and behavior. Um, Gabe Kaminer, I'm not sure if he's there. I think he's in the audience, but he's our information manager. I'll talk a little bit more about that. Mike Ruge, who is there, has been our program manager for over 22 years. And Mike celebrates his 25th anniversary with FIU this year. So there's lots of 25s to celebrate. And then Nick Ohm is our education outreach coordinator who also did his master's at FIU and is currently doing his PhD with Dr. Sarah Eddy from University of Minnesota and myself. And Nick does a lot with research for teachers as well as research experience for students. So I mentioned a few of the new collaborators. I also just want to give a call out to Dr. Jay Sa, Anna Simonson, Stephanie Wakefield, who's at Florida Atlantic University. We have collaborators new to our FCE5 who are from University of Miami, University of Florida, the Everglades Foundation, Yale University, North Carolina State, University of Chicago, and the Miccosukee Tribe of Indians of Florida. You're probably well aware of your new student e-board, but I just want to say thank you to James Sturgis, the president, Katie Johnson, the vice president, Mackenzie Zapata, the outreach coordinator, Sophie Costa, the treasurer, and Alia Jones, the secretary. So you're in great hands, and I'm looking forward to seeing what the student group does this year. And thank you all for what you do. So one of the big pieces of FC5 we term co-production. And that term, the word co-production is being reconceived because it's something that we all do a lot of through collaboration. But what we are referring to is how we deliberately engage the local, the tribal, the agency, the public partners, and how we utilize our long-term data to allow more buy-in, more engagement of the consequences of change in South Florida to enable the whole region to think about what are the risks, what are the adaptations, and how can we actually be a resilient coastal system that is undergoing rapid sea level while also being restored. A lot of the things that we're going to be building on in FC5 have to do with experiments and adaptation. Okay, so Marty Downs, who runs the LTR network office at UC Santa Barbara, she asked if I could share this slide, and this is to orient y'all to the network of LTR sites. So there are 26 long-term ecological research sites. We call them capital LTER. They span diverse ecosystems, as you can see from this map. There are 4 sites in Alaska. There's Moorea Coral Reef in French Polynesia. There's 2 sites in the Antarctic. There's coastal sites, there's urban sites, there's freshwater, terrestrial, and forested grassland sites. So there's a mix of different types of sites. The LTR network has been around for 45 years. There's over 7,000 data sets and 27,000 publications. And we're a network of thousands of people, thousands of scientists. So if you want to scan this QR code, and I'll save this presentation for folks for later use, you can join the network newsletter and get a sense of what's happening in the network, as well as where FCE fits in the network. And there's a link to that network page there as well. I want to just counter that by saying here's some stats from your FCELTR. So we have 14 core sites that span marsh, mangrove, and seagrass meadows. We have 25 plus years of data. We have more than 220 data sets that are publicly accessible. We have more than 800 publications. You can scan this QR code to take you to our story map that'll give you a sense of what's happening at each of the sites, at each of these core sites. Of course, we have lots of other measurements that are made in what we call satellite sites, and there's literally thousands of satellite sites that people have been collecting long-term data in that we are using in FCE5 to assess relative site representativeness, how the changes at our long-term sites are comparing to these other locations. Um, you can look at our YouTube page. We've got some videos there. I'll reference a few of those videos, or at least one of those videos later. But join the student group and join a working group. So I'm going to talk to you all about some of the working group um, opportunities and what the working groups are in FCE5. But let me just quickly go to, am I still sharing screen?
Yep, looks good, John.
Butte. Okay. So if you haven't yet joined as a member, you're going to go to this page that's on the website called Get Involved. So here's our FCLTR web page. When you're on the landing page, it'll look something like this, and you navigate down to um, or about, and then get involved. So that's how I got there. Um, Collaborators, affiliates, students, staff. For students, you click on that, subscribe to the newsletter, visit the student page, which maybe the student group, the student e-board has oriented you all to this already. There you can see student group at the All Scientists meeting last May. Um, your officers, and then events. There's so much happening. And we also publish upcoming events in the newsletter. What I want to go back to is member responsibilities. So when you join FCE as a member, you are agreeing to abide by the code of conduct, and I'll talk a little bit more about that. You're agreeing to our data policy, and I'll talk a little bit more about that. You want to make sure that your name is on our Everglades National Park research permit, that you complete the emergency contact form. So all these things that are listed here are really important for you to orient yourself to with time. And if you have any questions, you can reach out to myself, to Mike Ruge, our program manager, or to any member of the e-board. But really what we expect, which is listed here below, is for you to begin to collaborate and to publish your data and follow those data policies and help us build the long-term research. And I won't go into these specific statements, but I want you to read them and reflect on them and embrace them because that is who we are. That is who FCE is as a community. Let me go back. Oops, oops, oops. Okay, cool. By the way, feel free to interrupt me if there's any glaring questions or something's not looking right. Okay, so Gabe put together this data publishing workflow, and I wanted just to share it here to give you a sense of the steps that go into publishing data. And a lot of our students will ask, "Oh, should I publish data before I actually publish my paper?" And so sort of the ideal is that, well, yes, you would publish your data and then you could cite your data. They would have a DOI and your data itself are a publication. And then we actually can track the use of or the citation frequency of those data. So you cite it in your paper. Maybe you have 5 data sets. You cite all 5 of them, and you can cite them if they're published. You can't cite them if they aren't published. And you might say, well, why would I publish my data not just with my paper? And the answer to that is well, it's more complicated. You first off um, don't want to have people have to go to your paper only to find the data. So that limits the searchability, which also limits the use. If people can't find the data, if it's buried in your paper as an appendix or a supplement, then it's not as accessible for people. So the risk, of course, is well, if I publish my data, then someone might scoop me. And so what we at FCE do is we can deprioritize publishing student data, but we can't not publish it. We are obligated by the National Science Foundation, by the federal laws of the current federal laws to make publicly funded research, publicly funded data publicly available within 2 years of collection. And so the short answer is it's very rare for somebody to come in and take your data and publish on them. And I guarantee if they do, it's going to be a bad job. And so I want to encourage you to get in the mindset of publishing your data first and then citing it in your paper and then publishing your paper. Okay, I hit that one over the head, but why I do that is because it also puts you in the driver's seat of building your data publication, your data and metadata. So Gabe is always available, has office hours throughout the school year where he's makes himself available for consultation. He also offers workshops. He can also be consulted on an individual basis, but the best thing to do is to ask for his assistance before you need the data published. So he'll guide you in best practices, which, by the way, are also on our data page on our website. But he'll orient you to how the format of data need to be. There's this process called Easy EML, which is through the Environmental Data Initiative or EDI repository, which makes it really easy to correctly create metadata. It's not fast, but it is very accurate and comprehensive, and it takes a lot of the burden off of the the person, the author. We used to have to do all of this in a really cumbersome spreadsheet template, and now Easy EML is an online tool within EDI that helps create the correct metadata. And it makes it so that your data are then more findable and more consistent with other types of data. So once you go through this process of preparing your data and getting it ready to submit through Easy EML, it then comes back to the to Gabe, who checks it over, does a final review before staging it, and then publishing uh, the data. And then we make our data accessible through the LTR, the FC LTR data catalog, as well as through EDI. So it's findable both ways. So when in doubt, publish your data early. Okay, let's get into who we are as a long-term ecological research program. And I'm gonna, for a second, stop share and reshare because I want this to be full screen. Oh, hey, I can see the room. Hey, everybody. Okay, let me reshare and then, wait, why is I don't know why I'm not able to, oh, it's fine. I just wanted it to go into slideshow mode, but it seems to not. Hold on.
Does anyone have questions for John while he's pulling that up? Anything that you guys want to ask about the FC?
That's okay. It was when I went to there we go. Okay. All right. So I want to just briefly say a land acknowledgement. So the Florida Coastal Everglades is conducted on lands that were cared for generations by original peoples, and we honor that. And we acknowledge that that legacy of human occupancy from non-white people, indigenous people who have sovereign rights to the land is very powerful and it's an opportunity for us. So the Miccosukee tribe of Florida and the Seminole tribe of Florida have sovereignty. And in 2009, there was a US apology to Native people that enacted a reconciliation and an opportunity for healing, collaboration, and environmental conservation. And what is so cool about this is that we have active and willing, engaged partners in the Miccosukee tribe, and they have been a part of our science for a while. But that partnership has been officially strengthened. We have a representative of the Miccosukee tribe on our internal executive committee, and we are actively working with the tribe in research projects in the Everglades. And they have been a vocal transformation for how Everglades restoration, um, how it's affecting them, both positively and negatively. And they've been helping guide restoration, just like the science that we do. So we're a big collaborative LTER. We have more than 35 partnerships from academic institutions, state and federal and regional county level agencies and organizations, um, non-government organizations and private foundations, public schools, and of course our tribal partners. So the responsibilities and expectations I mentioned, the code of conduct, it's linked directly in that responsibilities page. So when you go here, the code of conduct is here, and I've actually linked it into the presentation as well. But and I want you to read that, but I also want to just say, look, here's how we operate. We're about safety, we're about openness, we're about respect, we're about being ethical, both scientifically and in terms of social ethics, and we're about collaborating. And if at any time there's a problem, you can report concerns to your supervisor, to myself, or any member of FCLTR leadership. You can also, and this information is in the code of conduct, you can also report at report.fiu.edu. And if you're not from FIU, you can report through your home institution, and this can be done anonymously. So our goal is reduce, to eliminate or not have any problems, but of course problems arise. And if you need help, we want to help. And here's some pathways for you, for all of us. Okay, let's get into where we play. So, this is a map that Mike Ruge generated a couple of years ago, and it's really beautiful. So it shows you the two main drainages in Everglades National Park. So we work in Everglades National Park, which is this boundary, this dashed boundary in South Florida. So Shark River Slough, which drains beneath Tamiami Trail through now more recently bridges that have occurred along the trail. These marshes look like this over here on the right-hand side. They're freshwater marshes. They have a ridge dominated by sawgrass. They have open water sloughs that are oftentimes filled with periphyton and submerged aquatic vegetation. So Shark River Slough drains down into the Shark River, which is down here by SRS 4, 5, and 6. These are riverine mangroves that are tidally influenced from the Gulf of Mexico. So you see this green arrow is the flow of marine water, and the blue arrow is the flow of fresh water. So there's a lot of water mixing and exchange in the Shark River estuary, which is the Shark River. And we have extremely productive mangrove forests in Shark River, some of the most productive ecosystems in the world. Taylor Slough, which starts along the eastern boundary of Everglades National Park and is fed by the C-111 canal. It is a marl-dominated short hydro period, although it's now currently perennially wet with restoration, but traditionally it would dry down for months out of the year. It's a much lower productivity marsh. It's not peat accumulating, it's marl accumulating, and it segues down into the area in Lower Taylor, which is an ecotone that has connectivity with groundwater. And further down, we have scrub mangrove forests at Taylor 6 and 7, which empty out into Florida Bay. And Florida Bay, we have a transect from the northeastern part of Florida Bay to the southwestern part. And you can see these marine water supply arrows. We have less marine influence further in. This is more protected part of Florida Bay and more direct connectivity with the Gulf of Mexico further to the southwest. Florida Bay has some of the world's most I think it has the world's most contiguous area of seagrass meadows. It is an extremely low productivity environment like Taylor, but even more so. And what we have in these in the Everglades are these gradients. So we have freshwater to saltwater gradient, and also concurrent with saltwater gradients, we have a phosphorus gradient. So where we have larger green arrows to smaller green arrows, the larger green arrows have more marine sources of phosphorus. We're a reverse or upside down estuary. So the marine environment stimulates productivity and biomass in our downstream coastal environment relative to our interior marshes. So FCE5 has a conceptual framework and it's quite detailed, but I wanted to show you it to orient you to the science behind our current program. So we have 5 focal areas: climate variability and change, hydrologic presses and pulses, disturbance legacies, coastal social ecological landscape, and resilience and trajectories. And our framework, our conceptual framework is such that we know that climate variability influences hydrology in South Florida in complicated ways and in ways that are different from the rest of Florida. So in South Florida, our rainfall is actually higher than in other parts of the state of Florida, and they're driven by different factors that I won't get into in this slide, but I will a little later. These hydrologic presses and pulses come from fresh water in blue and marine water in green from different factors. The freshwater presses and pulses are both restoration, storms, where y'all have been experiencing flooding recently as a freshwater storm pulse, seasonal rainfall. Those storm pulses are greater in the wet season than they are in the dry season. The marine saltwater intrusion is a continuous press, but we also have storm surge pulses. And that saltwater intrusion press is exacerbated by rising sea levels. So, together, these fresh and marine hydrologic presses and pulses can leave legacies of disturbance on the landscape. There are areas that have dried down, areas that have been intruded by saltwater, and areas that are being restored by fresh water. And we want to know how those presses and pulses of marine and fresh water are affecting vegetated coastal wetlands. And so this smaller illustration here is a expectation of systems that have high fresh and marine water pulses, or basically have a lot of hydrologic connectivity, are going to maintain vegetated coastal systems. Those that either have low pulses or high presses are going to shift to more of a open water or eutrophic state. And so that matters because in South Florida, elevation is everything. And if you are vegetated, you're building elevation. If you are losing vegetation, you are also losing elevation. And that is of concern not just to the marsh, mangrove, and seagrass systems of South Florida, but to human urban centers. And so through the way that we apply fresh water or use fresh water in our daily lives, how we value the systems on the landscape, whether there's enough fish for recreational fisheries, whether people care about having mangroves and are willing to protect them as opposed to remove them, and whether we value the marsh and its native species relative to its invasive species. All of that influences the functioning on the landscape, the consumers, the vegetation, the decomposition and proliferation of microbial biomass, and whether or not we are building in the yellow, staying stable, or declining in the red. And so in FCE5, we're saying that all of these systems, urban, marsh, mangrove, seagrass, they're all affected by the way that we use and push and pull water across the landscape. And that collectively that drives our economy, but it also drives the ecology of South Florida and whether we're going to continue to grow or whether we can keep going or whether we are dipping down. And so we recognize that the urban or the human and non-human components of the landscape are interconnected. And these terms guide, I won't go through them, but this is how we use terms to define disturbance, the legacies, the presses, the pulses, and the resilience. So this framework is how we organize our research question, our central research question, which is how do disturbance legacies interact with increased hydrologic presses and pulses and their associated subsidies and stressors to influence long-term resilience and trajectories across spatial scales in coastal social ecological systems. It's kind of a mouthful, I recognize. And we have some predictions based on these 5 focal areas. So we predict that disturbance legacies will constrain social ecological responses to increasing hydrologic presses and pulses from human and climate drivers. We also predict that climate variability and change are increasing regional climate forcings like temperature and the distribution of water in the Everglades, and that's going to continue to alter the timing and magnitude of seasonal hydrology. We predict that increases in hydrologic presses and pulses will enhance connectivity to both abiotic and biotic resources to stabilize or stress this oligotrophic or low productivity ecosystem. In the coastal social ecological landscape, such as ours, water governance, how we use water, the cultural and economic values around the natural environment as influenced by water interact with ecosystem structure and function to influence elevation, carbon fluxes, and ecosystem services. And lastly, resilience and trajectories are influenced by the integration of functional redundancy in the landscape and how that affects ecosystem processes, social ecological feedbacks across time and space. So I want to pause to ask if there are any questions before I dive into some of the working groups, who's running them and what they're testing. If not, carry forth. Okay, so question one in working group hydrologic or climate variability and change working group is how are increasing temperatures and altered timing and magnitudes of precipitation interacting with disturbance legacies to drive urban and wild land climate feedback. So basically what's going on in the Everglades itself and how is what's happening in the urban environment influencing what's happening in the Everglades in terms of climate. So the hypothesis here is that increased air temperatures will reduce the duration and increase the magnitude of seasonal rainfall pulses. And this is essentially looking at climate mean and variance. So this is probability of occurrence and the bell curve under our normal scenario or previous climate scenario, and then the new climate shifting to a warmer environment. We know that temperatures are increasing, so we're going to see more warm temperatures, a higher mean temperature, but also the variabilities in temperature, both highs and what's not shown here, but lows are occurring with climate. So we do get cold snaps in South Florida. They do drive lots of disturbance legacies in the system. But in general, we're seeing increasing high temperature anomalies. And so if we have both occurring where we're shifting the mean and increasing the variance, how do we deal with that in terms of planning? And then I also mentioned that this group is focusing on the urban influence. So the urban environment is heating up obviously faster than the Everglades, but it's causing higher rainfall in the Everglades because it's warming the atmosphere, drawing more moisture out of the land surface as well as the ocean surface, and causing increased rainfall intensity. So this group's led by Rob Bergman here at Earth and Environment and Jayantha Abhisakura, also in an Earth and Environment. Dr. Amy Clement and Ben Kurtman from University of Miami, and Rigoberto Oliveira is Rob's student, soon to be, I think, postdoc. So this working group number two, hydrologic resources and stressors, and by the way, these color-coded numbers go back to the five themes: legacies, climate, hydrology, landscape, trajectory. So the color-coded numbers are suggesting that these working groups are underneath those themes. So in the hydrologic resources and stressors theme, we're underneath the legacies as well as the hydrologic presses and pulses. And so our question is how are amplified hydrologic presses and pulses and legacies of disturbance changing the spatial and temporal variability in water chemistry in the surface and in the groundwater? And our hypothesis is that increasing hydrologic presses will shift long-term sources of water and composition of dissolved organic matter, while increasing hydrologic pulses will alter the seasonality and magnitude of carbon and nutrient concentrations in the water. And this working group is led by myself and Shimon Dewinski and Chris Osborne, and then I just have Rafa, because he's also part of the working group and our main LTR, FC LTR technician, along with Emily Standen and Sandro Stumpf, who maintain this long-term water quality data. But the figure I show here is just it's from all of our ecosystem types in Shark on the left-hand side, marsh, ecotone, mangrove, and Taylor marsh, ecotone, and mangrove, as well as Florida bays, seagrasses. And you're seeing the blue line is the long-term trend in water level relative to the monthly variability, monthly and annual variability, seasonal variability. Everything is increasing in water level. Water levels are going up from restoration as well as sea level rise. So we know that we're seeing this upward trajectory in increasing, definitely increasing presses and to some extent increasing pulses. The vegetation working group is led by Dan Gann, Jay Sa, Johannes Kraus, and Evelyn Geiser. And that working group is asking how do increases in presses and pulses interact with disturbance legacies? We all start our questions the same to affect the composition, distribution, and productivities of macrophyte, microbial mat communities and their ecological functions across the landscape. And so one of the hypotheses is functional resilience is a product of stress resistance, functional synchrony, dispersal capacity, and functional redundancy of communities among connected patches controlled by connectivity at multiple levels. And here's an example from Jay Saw's group where he has these long-term transects that go from the eastern boundary along Taylor River, the marl-dominated transect or drainage, and he moves along the edge of these marl prairies, which are higher elevation, into the slough and ridge habitats. And has he has thousands of long-term points that he samples every 2 to 5 years along these transects and has been doing that for decades. And um, his lab is looking at functional richness and functional divergence as a function of changing mean water depth. And it's not the same for both for all transects. So there's a lot of spatial variability. And one thing to note is that functional richness in M1 transect, which is a shallower water transect, generally shallower water, is increasing with increasing water depth, whereas functional richness in the deeper water M4 transect has not changed, or M2 transect has not changed with increasing water depth. But functional divergence is decreasing with water depth in both transects. So this is just an example from Carlos Politos's dissertation work with Dr. Sa. The consumer working group, which is led by Dr. Rihej and Santos, as well as Dr. James and Dr. Rezik, as well as the Height House group, they've been looking at how presses and pulses and legacies of disturbance influence the reliance on green energy pathways and the change in resource quality from subsidies across the landscape and how that's affecting the structure and function of food webs. So they have more hypotheses. I'm not going to read through every single one, but the first one is about green and brown and how that's changing. So green being algal, brown being detrital. And greening is predicted to reduce the niche breadth. So consumers that are fed in a more green environment and a more productive environment may not have to search the landscape for other resources. So their isotopic niche is predicted to be smaller. Hydrologic pulses drive resource subsidies and affect where consumers need to feed in the landscape and what that looks like. And so I know this is one of James' figures, and it's an older one, I'm sure, and I'm not sure if this has been published, but is ready to be, and I know it's really been on top of mind of James for a long time.
In
Um,
review for the fifth time. So
yeah, get it done.
there.
Okay, and I know it's changed a little bit, but basically this is a figure from a dry season sample on the left and a wet season sample in Shark River Slough and Taylor Slough, and looking at the relative source contributions in the marsh, mangrove, ecotone, and the estuary river delta, and then out into the bay in Florida Bay. And without going into the details of this, just to say that this is complemented by this prediction that over time we're going to see a proportion of higher green energy, a lot of that coming through increases in marine contributions, as well as changes in the distribution of phosphorus in the landscape, and that trophic niche width with proportion of green is predicted to go down. And there's some evidence from the research that they've collected above that is showing this, and that the niche size is also going down when there's more sunfish biomass. So when there's more food, there's less need to forage the landscape for more food, and the niche size goes down. So they've been showing that this is variable year to year, because the way that sunfish biomass occurs deals with both temperature and water depth in the marsh. And so that varies year to year. And for example, this year, marsh water depths were really low. So next year, sunfish biomass will be predicted to be low, and snook niche size should be high or higher. So question 5, we got 7 total questions. So we're at question 5. This is the detritus and microbe working group. And we are looking at we're looking at disturbance legacies in the social ecological landscape and the effect of that on trajectories. So how are increasing presses and pulses of fresh marine water affecting organic matter processing and microbial community functions in wetland ecosystems that have been exposed to different legacies of disturbance? So we predict that the presses and pulses are driving soil anaerobic conditions and functional redundancy in wetland microbial communities, and that disturbances that increase nutrients, like hurricanes that bring in subsidies of phosphorus from the marine environment, saltwater intrusion, which generally stress the environment, but also bring in phosphorus subsidies, and fire, which is a very prominent feature of Everglades management, where marshes are burned on a regular fire return interval. Fire is a release or a pulse of phosphorus. So these increases in nutrients will reduce functional redundancy in microbial communities, making them more spatially and temporally similar, and enhance microbe-driven organic matter processing rates. So basically driving down the driving decomposition rates and reducing storage of organic matter, which can contribute to changes in elevation. Well, this is just an example of some work from this working group is co-led by Will Martin Sabena from University of Florida, Anna Simonson, who is at FIU but also now relocated to University of Alberta, myself, Chris Osborne, Nathan Wisnowski at Mississippi State University, and Jian Cha at UF, working with Will Martin Sabena. So this came out of Wilm's lab and showed that the freshwater marshes that have been exposed to salt have an increase in methanogens, in the gene abundance of methanogens, whereas brackish water marshes have higher sulfate reducing microorganisms and are also influenced by saltwater intrusion, or saltwater dosing. And so the ground and surface water, fresh or marine, um, exposure of soils can change the expression and the abundance of microbes that are either in a methanogenic or a sulfate reducing environment. And as we have real anaerobic marsh conditions that are freshwater that get exposed to salt water, we may be um, transforming them into environments where microbes are primed by changes in redox to decompose more and flux more carbon out of the environment. So we're trying to investigate that across the landscape. So I want to, there we go. That's better. Okay. The sixth question is the Ecogeomorphology Working Group, which is led by Dr. Troxler in Earth and Environment, Dr. Kandakar Ishtiaq, who's now at South Florida Water Management District, and Dr. Sklar at the district, among others, but those are the three major working group co-leads. And they ask, how do increases in marine and freshwater pulses and presses interact with disturbance legacies to drive the fluxes and net ecosystem storage of carbon that influence the resilience and trajectory? So they're really focusing on the surface elevation changes. So the hypothesis is that hydrologic presses and pulses increase carbon fluxes more than carbon storage in wetlands with disturbance legacies. So if this system has had a disturbance legacy, it's going to be negatively affected in terms of carbon flux being going up relative to being stored. And so this little graphic in the lower right, you saw this press and pulse diagram from our conceptual framework. And here below, it's an exposure plus sensitivity minus adaptive capacity equals the vulnerability. And so wetlands that are exposed to disturbance and to presses and pulses, shifting presses and pulses may be more sensitive and have less adaptive capacity. The final working group, which is trying to tie in a lot of data across our working groups. Question 7, our social ecological landscape. They ask, how have these legacies and climate impacts and restoration together changed the perspectives of ecosystem services and decisions that drive social ecological resilience in urban communities? Basically, have people paid attention, and are they changing the way they think and the decisions that they make? So the hypothesis is that understanding how disturbance legacies, climate change, and restoration interact to influence services and societal resilience, social ecological resilience, it's enhanced through stakeholder co-production of science. So basically, when we don't just do science for ourselves and then hand it over, but rather we invite people to work on the science with us, that science gets used, it gets communicated, it gets co-produced. And that is really, we feel, the pathway towards real understanding about risk, adaptation, and decision making. And so this is a little snippet from this working group. Uh, it's not fully showing you the full diagram, but it's just saying, currently or in the present, we're going to use co-production of knowledge and design for integrated cultural and economic values of ecosystem services. So we're not going to just take the ecology and the social and separate them, but we're going to integrate them into this landscape where they interact. And here's some of those examples of partners and co-producers. So this is being led by Stephanie Wakefield at Florida Atlantic University, Kevin Grove here at FIU, Mahadev Bhatt, who's an economist here in Earth and Environment at FIU, Meenakshi Chava, who is also an economist at the Everglades Foundation, Neil Brenner, Alexander Arroyo, and Gurga Basic from University of Chicago. So I just want to give a shout out to some of the cool things that use of technology such as machine learning and AI are starting to um, allow for us to do more informed and more predictive work. And so this work here on the right-hand side is by Boya Zhang, who was a graduate student here with Shimon Dewinski and is now in his second postdoc. And I'm forgetting, I think he's at Yale now. He was at UC Merced and now he's up at Yale. And Boya was using machine learning to look at, and this is a paper that came out last year in remote sensing, to look at the um, looking at our plot level data relative to Worldview 2 satellite images and scanning to look at changes in canopy post Irma. So Hurricane Irma hit in 2017, and we've got these long-term, these little plots here on the bottom. We've got these long-term data sets that we can use as reference points. So he was able to hindcast pre-Irma canopy and look at post-Irma canopy and then the change. So where we saw the greatest declines, and those greatest declines were along the coast and associated with Shark River. And so these are the different transects that were run, and just looking at the different species of mangroves that were lost, and just a real high power way of looking at a lot of information and taking long-term data and scaling it up. And Hamadri Biswas and Dangan are doing something similar with red mangrove expansion in Taylor Slough in the southeastern saline Everglades. So Taylor Slough empties into Florida Bay, as mentioned before and shown here. And Hamadri is using a really fine spatial resolution, 5 by 5 meter resolution, to show that red mangrove are infilling with sea level rise, but also red mangrove are infilling and expanding with fresh water coming down Taylor Slough. So just to say that we're using these tools and we need to continue to use them. Um, I got this note from Gabe the other day that we're using AI and GitHub Copilot to work through repetitive code and check data format consistency. And then I put this question, how can we use AI to integrate across ecosystem models? And I think that's where we need to begin to continue to push. So these are just two examples. Also, I mentioned that we are using experiments. Experiments allow us to test mechanisms. So Kelsey Solomon and Evelyn Geyser have built these Western Everglades phosphorus flumes that are out in Big Cypress. This is one photo of the flumes that were being put out at one of the 4 sites. And they're dosing at different concentrations of phosphorus to try to look at different phosphorus uptake pathways and capacities and really get an assessment for um, how these oligotrophic marsh habitats in more forested ecotones, how they're responding to fresh water that's at various concentrations of phosphorus. And we can't do these types of experimental manipulations everywhere. They're very limited, and so when we do them, they're really powerful. And this other example is Mimi, the mangrove experimental manipulation exercise, which has been built out. You can see from Google Earth the footprint, the boardwalk, similar to the boardwalks here, but you can see them here along US 1, crossing over the C-111 on the way to Key Largo. And this is a multifactorial manipulation of phosphorus, mangrove, and sediment addition to really look at those effects of interactive drivers on elevation change. And so some pretty pictures from Tony Slayman. There we go. We are driving ecological discoveries and you are driving them. I just love that picture. Um, Join an FC5 working group. Reach out to anyone in the working group. You can find the contact information and more information about working groups on our webpage. If you have questions, ask me, Mike Ruge, or anyone else, but connect your work to a working group. It's a really powerful way to collaborate and also to get co-mentoring and just to be a partner. Know that you're contributing to the science of Everglades restoration. Everglades restoration is the world's largest ecosystem restoration project ever. It's accelerating and it is needing us to guide and inform how it's going and where it needs to go. I mentioned before Nick Ohm, our education outreach coordinator. Um, you can contribute to education and outreach. Many of you already do. There's, 150 of you at any one given time, and we have more than 10,000 public school students that we reach, and like 30 some teachers in Miami-Dade County who we've trained who are eager to have you be a participant in their classroom. Nick and I are in the process of putting together a workshop which will allow students to get workshop credit to do the things that we would like you to do and you want to do for education and outreach. So stay tuned for that, and thank you. And then lastly, building data for synthesis and transparency and public access is really critical. Again, we have over 220 comprehensive data sets. Thanks to Gabe Kaminer, those data sets are QA QC'd, and we are building them better, and we're relying less on Gabe because he's training us on how to do best practices. But that is so, so fundamental to who we are as a long-term ecological research program. And I encourage you to be a part of that. Okay, so last but not least, we've got this beautiful video. This is our FCLTR All Scientists meeting this last May, our 25th anniversary. And you can also find this video on our YouTube, but Tony put together uh, this beautiful video that I hope you can hear okay. And here we go.
Audio is a little shaky, John. We're actually not really hearing it right now.
You can't hear it? Did you
It
say?
came through for a bit, but sounded a little like mumbled, and then it's not playing right now.
Okay, can I ask you if you can, well, you know what? Do y'all know how to find it on the YouTube? Maybe if I just go to the YouTube.
Or you can send me the link in chat and I can just pull it up on this computer.
Yeah, I'll do that. Okay, but let me send it in chat. Go for it. I'm going to stop share. I think I see quite a few new faces. I wish I was there in person, but I'm glad y'all
Do
are.
you want me to share it with
Yeah, go
everyone?
for it. Yeah, go ahead. I'll be quiet.
Got that?
Can you reverse it? Cool. I hope you all enjoyed that video and you're a part of us. So, like it or not, here you are. If there's any questions, I'm happy to take them, but also I'm sure you're probably happy to potluck. But
Yeah, any questions for John? Well, John, thank you so much for giving that presentation. It's a lot going on in the FC and a lot to organize, but you condensed it well. And I think we have a lot to look forward to in the future with FC5.
awesome. I'm really looking forward to meeting those of you who are new and keep on the horizon that we're going to have an all scientists meeting in May. We have that at the Fairchild Tropical Botanic Gardens, and that's where students present posters, and that's where you meet more of these scientists. But have fun, be active, ask me where I can help you, and welcome.
All right, well, John, we're going to give people time to get food and then we're going to do our presentation.